Welcome everyone. Today we are going to learn about passive transport by channels. Now at first I want to recapitulate what I have already mentioned in my previous video, basic facts on transport, that there are two types of movements occur. One is downhill movement. Downhill movements can be understand but when someone actually come down from a hill, it feels easy. So here molecules or ions transported down their chemical concentration gradient, that means from higher to lower concentrations, and this process is thermodynamically favorable. That means there is no requirement of external energy, and that's why it's called passive transport. As you can see, the molecules that are transported are actually higher. Let's suppose this is the outside and this is the inside of the cell, so higher outside than inside, and here the molecules move down their concentration gradient. That's why it's called downhill movement. But there are three different movements occur: simple diffusion, channel mediated, and transporter mediated. Here I will actually mainly talk about channel mediated, but at first I want to mention that in case of simple diffusions, no requirement of transport proteins is there, because molecules actually directly traverse or cross the lipid bilayer of the plasma membrane. Now here the movement of these molecules, like I have already mentioned in my previous video, that plasma membrane is permeable. Some molecules like gases, and as because this lipid this plasma membrane is actually lipid in nature, that means it is hydrophobic in nature. So hydrophobic molecules like steroid molecules or fat soluble molecules are actually readily cross the lipid bilayer or plasma membrane. And some small polar molecules like urea, ethanols also actually cross the plasma membrane. Now here what the rate of the simple diffusion depends on three important points. One is chemical concentration gradient. That means as much as the chemical concentration gradient is present, the more it will rapidly enter the cell, or actually move into the cells. Another is called hydrophobicity. So the rate of simple diffusion depends on hydrophobicity, because I have already mentioned, as because these molecules are moving through the hydrophobic environment. So more the hydrophobic the molecule is, it more it can actually rapidly cross the bilayer. And also the size of the molecule depends, because as much as the size is small, the molecules can actually enter inside the cell or outside the cells. Both can occur. Now, today we are actually focusing on channel mediated uh, passive transport. So let's talk about channel mediated transport. Now, this signs here at the triangle signs denotes that the base of this triangle denotes the higher concentration of the ions or molecules than the inside, because it is the uh, sharp point of the triangle that shows the lower concentration of the same molecule. And we know that channels, through the channels, molecules or ions actually move from higher to lower concentration. That's why it's called downhill movement. And the rate of movements of ions is very high, 10 to the power 7 to 10 to the power 8 ions per second. Okay, now channels can transport water, ions, or hydrophilic water small molecules. Now hydrophilic means water loving small molecules. It shows downhill movements and does not require energy. So the molecules actually move from higher to lower concentration gradient. Okay, now one important point we have to understand here for channels, that ions or molecules that carry a net charge. Okay, that means it carries, if it carries a charge, it, or whether it is positive or negative, its movement across a membrane is influenced not only by the concentration gradient, but also the membrane potential or electric potential, that is called voltage, and togetherly we call it electrochemical gradients. Now let me explain. Suppose this is the first situations where the this is the outside of the cell and this is the inside. And if this is the first condition, here as you can see, only the concentration gradient matters for the movement of the molecular ions, because there is no membrane potential. So in this case, the molecules or ions actually move only by the concentration gradient, depends on the concentration gradient. Now if this is the second situation, what will happen? let's suppose there is a charge separations across the membrane. That means here positive charge or ions are more than in the outside than the inside, so inside is negatively charged, so there is a electric potential or membrane potential is present in these cells. Also there is a concentration gradient present. That means outside the positive ions are more than the inside, then the movement of these positive ions will occur more than the ions that depends on only the concentration gradient. That means here the movement, as you can see this green arrow sign showing more or rapid actually movement of molecules or ions, because here both the chemical concentration gradient and the membrane potential actually helps to move these molecules. As you can see here, because the inside is negatively charged, so positively charged molecules will be, or positive ions will be preferred to enter inside the cell. So electrochemical gradients with a membrane potential. But what will happen for the negatively charged molecules or ions? You will see that the rate of movement here is very low than the concentration gradient also, because here the negatively charged ions are actually moving from outside to the inside, and the inside charge is actually negative. That's why it is less than the concentration gradient also, because here also the negative charged molecules are present higher in concentration outside than inside, but the as the membrane potential actually favors the positive charge, not the negative charge, so the movement or the rate of the movement of these negative charged molecules or ions will be lower than concentration gradient. Now these membrane potentials, that means the more positive charge present outside, basically in almost all plasma membrane or cells, the electric potential across them is that with the negative inside is usually negatively charged and then the outside. Now this membrane potential is actually established by ATP powered pumps, and this is an example of ion gradients across the plasma membrane of mammalian skeletal muscle cells. As you can see, the extracellular concentration of the ions like sodium, potassium, calcium, and chlorine, these main ions, let's suppose this is the outside of the cells, is that in case of sodium, the concentration is outside higher than inside. So what will happen if there is any channel opens? The sodium will come inside of the cell. In case of potassium, the opposite will happen, because in the intracellular or inside of the cell is the concentration of potassium ions is higher than the outside. And now this, I want to mention that all these differences are actually established by ATP powered pump. Okay, then the channels actually utilize this electric potential and also the chemical concentration gradient of the ions. In case of calcium, the concentration is higher outside than inside, so it will move inside. And in case of chlorine also, it will move inside. But as I have mentioned, negatively charged ions are actually prevented to move inside, because already there is negatively charged molecules are present inside of the cell. Okay, now let's talk about channels in more details. Channels could be different in nature. One is called voltage gated channels. It means these channels depends on the membrane potential or voltage change of the membrane potential. So as you can see, it is closed when the membrane potential actually changes. Like here outside is plus, now when it actually becomes negative, then the channel is open, so it is voltage gated channels. That means the opening of the channels depends on the voltage. Another kind of channels could be ligand gated channels. Here the ligand could be from outside or inside, so ligand gated channels. And mechanically gated channels are also present in the cells. So some channels are open much of the time. This is called non-gated channels. There is no gating strategy, but most ion channels are open briefly in response to specific chemical or electrical signals. Like these are called gated channels, and it close again. So there is opening and closing of the channels. And one thing we have to understand, that with prolonged stimulation of most ion channels go into an inactivated state, and this is called also called desensitized state of the channels. Here what happens, these channels becomes refractory to further opening until the stimulus has been removed. So this is called inactivated. So in case of gated channels, there is three conditions. One is called open, then it actually becomes inactivated, then it close. Okay, so this is all about passive transport through the channels. In my next video I will talk about uniporters as a passive transport.